Michael Levin is a developmental biologist working at the Tufts University Centre for Regenerative and Developmental Biology. If you haven't been introduced to Michael's work yet, prepare to have your mind blown. Many things I thought I knew about biology have now been overturned by Michael Levin's studies relating to growth and form in biological organisms. The discoveries made by Michael and his team may be just as big a deal as the discovery of DNA. In this episode, we'll cover Michael's discoveries regarding growth and form and their implications, including potential cancer treatments, regeneration of limbs and other body tissues, including brain tissue, and the development of novel organisms via bioelectric methods rather than by directly altering DNA. Michael Levin, welcome to the podcast.
Well, thank you so much. I'm happy to be here.
Awesome. You've done experiments on flatworms called planaria, an organism that can regenerate a full body from a body part that's been cut off. What did you learn about memory from those experiments?
Well, there are two kinds of memory that we worked with. We worked with behavioral memory in three-dimensional space, which is what most people think of as learning and behavior type of memory. And then we also worked on the memory of regeneration, meaning anatomical memory. So with respect to the learning, we recapitulated with new modern automated techniques, something that was actually discovered by James McConnell back in the 60s, which is that if you train planaria and get them to have a particular kind of memory, in our case, we train them to remember that they would be fed with liver on a particular bumpy sort of shape on the floor of the petri dish. That then what you could do is cut off their head, which contains a centralized brain, and the tail would sort of sit there for about a week and not do anything, and then eventually it would regrow a new head. And when those animals regrew their new head, then you could test them behaviorally and recover the fact that they still remembered the original training.
That's
so this
incredible.
this is very interesting. McConnell published this in the 60s. Some people were not able to reproduce it. Some people were able to reproduce it. It was fairly controversial. And we, using an automated device to train and test the planaria, we showed that he was 100% correct. It absolutely works. And it really forces us to ask the question of what exactly is the substrate of memory? Where is it stored? And how, in particular, how does it move through the body? In this case, memories are literally imprinted on this new brain as it develops from scratch. And not only is that sort of some profound questions about memory and in general, but actually it has biomedical implications, because presumably in the next decade, we're going to have stem cell therapies where patients with 40, 50, 60 years of personality, of memory, and so on, are going to have new stem cells put into their brain for degenerative disease. And the question is, what's going to happen, right? What happens to the cognition of an adult body where parts of the brain are replaced by new cells?
So when you cut a planaria up, say that tail end that you were just mentioning before, does that actually have DNA in it, or is it somehow growing back and remembering that form without having DNA?
Well, every cell in the body of a planarian has DNA, so it certainly has DNA. But having DNA is a far cry from understanding how large scale shape forms.
Yeah.
Because if you think about it, if you take a planarian and you cut it exactly in half, the cells on one side of that cut are going to make a head. The cells on the other side of the cut are going to make a tail. Those cells were direct neighbors sitting right next to each other. They all have the same DNA, and yet they have completely different anatomical fates. So, that's something else that we can maybe discuss is this notion of what exactly does DNA do in terms of producing three-dimensional shape. But in any case, certainly the tail has DNA.
Okay. So if you had to make an educated guess, where would you predict the information for the body plan is stored, or how?
Well, the best way to think about this is using the hardware-software analogy. And I want to be clear, I'm not suggesting that living organisms resemble the kind of computers that we deal with on a daily basis, in the sense of being sort of linearly executing a set of instructions that were written by a human and all that. Of course, that's not what's going on. But there is something very instructive and powerful about this reprogrammability kind of concept that exists in computer science. What the genome gives you is what the genome specifies is the micro level hardware that every cell gets. So every cell has a complement of proteins. Those proteins are specified in the DNA. That's what's in the that's what's in the genome. If you read the genome, there's nothing directly in there about the size, the shape, the symmetry type, the structure of the body. You don't see any of that information. What you see is the micro level hardware of every cell. Now, that hardware is amazing because when that hardware is run in parallel fashion, meaning every single there's a group of cells. Every cell is running some subset of these proteins that are specified in the DNA. That hardware has some really interesting software modes, and we call that physiology. Basically, it starts the cells are communicating with each other, they're processing information, they're doing computations, and like any machine that you might build, it has a default set of behaviors that it does out of the box. So you can think about buying a calculator from the store. So there's a calculator, there's a hardware specification that describes exactly where all of the transistors and various other components of the calculator sit. Doesn't say anything about mathematics or any of the other things that calculator can do. It just says where do the transistors go. And then you put in some batteries, you turn on the juice, that's equivalent to sort of booting up the organism during development. The electronics starts to act, and it has a default behavior. It starts out and it shows a little 0 on the LCD screen. That is the default behavior of that piece of hardware. If you ask where is that 0 programmed, well, it isn't programmed anywhere. It's the workings out of the laws of physics, the laws of mathematics, and so on that are implemented by the machine that you just built. And then you might find out that it's a reprogrammable calculator, which means that it can show a 0, but it can also do a lot of other things, including store memory and do all kinds of interesting mathematical functions. Again, all of which you are not going to find on the hardware specification of the calculator itself. So. It's a tricky question to say where is the information for a body plan stored. It isn't directly stored anywhere. It is manifested through the laws of physics and the laws of computation by a machine, and that machine is specified by the DNA. But in manifesting those laws, something very interesting happens, which is that in addition to biochemical circuits, biomechanical kinds of properties, there are these bioelectrical events that occur in tissue, and these bioelectrical events tend to store a coarse-grained memory of what the large-scale structure is going to be. We can now read those memories, and to some extent we can interpret them and we can rewrite them. And that is, it's very much parallel to what happens in neuroscience, where there's a collection of cells. Those cells communicate electrically. Those are the cells in your brain. And as a result of various properties of computational networks and other laws of physics, they're able to store memories, guide behavior, and so on.
Yep. Wow. Okay. What are non-neural bioelectric states?
Yeah, basically, if you think about the fundamental commitment of neuroscience is that all of the mental states of you as a person or of any animal, all of the memories, the preferences, the goals, all of these things are encoded in some fashion. They're literally mediated by the electrical activity of your brain. So in theory, and people are working on this is called neural decoding. If we were able to measure all of the electrical states in your brain, meaning which neurons are doing what, and we knew how to decode this, we would have direct access to all of the things that were part of your cognitive apparatus. So if we sort of think about where did that trick come from of storing computation, storing the different kinds of mental states, plans of the future, memories of the past. How do you store that in electrical networks? Well, it turns out that didn't come from nowhere, sort of just evolved when brains came on the scene. Evolution discovered long before that electrical networks are great at computation and at processing information. And so somewhere around the time of bacterial biofilms, individual cells, and at that time bacteria, evolved the ability to join into electrical networks that process information. And if you want to know what information they're processing, it's distributed across the population of cells or of bacteria in a biofilm, and the electrical states are basically the patterns of electrical activity, the resting potential of the different cells, the ways that those potentials move and change, and so on.
Right. Can you tell us about your work with Danny Adams regarding the electrical face?
Yeah. Danny originally came to my lab to study the. She started out as a postdoc in my group, and then eventually became a PI herself. Uh, she came with the intent to really try to understand some of the the endogenous bioelectrical patterns that specifically dictate the gene expression and ultimately the anatomy of various organs. And she started with the face, and what she found was something quite amazing, which we called the electric face, which is that prior to the regionalization of the ectoderm that's going to become the face into all the different components of the face, right? So you have to demarcate where the eyes are going to be, the mouth, the various other organs, and so on. She found that there's a bioelectrical pre-pattern, that is before the genes turn on to really pattern that face. There are already patterns of depolarization and hyperpolarization that demarcate where all of those things are going to go. And more importantly, what she found is that not only can you read the picture of the future face long before it actually appears, that pattern, as she was able to show, is instructive. In other words, it's if you move those electrical states around. Let's say you wipe off the voltage state that's corresponds to building an eye in a certain location. You wipe that off the face, and you put that you induce that electrical pattern somewhere else, which we showed that years before that. You can induce eyes in various other locations in the body. In other words, the pattern of the face is the pattern of the electric face is instructive for where the components are going to go. And she was able to also use this to provide an explanation for some human craniofacial birth defects, because there are mutations in human patients, which, for example, like Anderson-Tawil, for example, gives you cardiac arrhythmias, but also facial dysmorphias. Right. And it was understood quite early why there's a cardiac arrhythmia, because it's an ion channel. It's a mutation in an ion channel. It was not understood why would the shape of the face change just because an ion channel is mutated. And so her work very naturally shows why that happens. It's because the electric pattern that normally underlies the design of the face is abnormal in those patients.
Right. It's interesting. In another fascinating experiment, you took the skin cell from a frog and found that despite being an ordinary skin cell, it could develop ability to move around in a tunnel and possibly make decisions, if I remember correctly. And can you tell us about that?
Sure. Yep. I think you're talking about what we call the Xenobots, and this is joint work with Josh Bongard's lab, and so Sam Kriegman and Doug Blackiston are the people also involved on this team. It's not a single cell. What happens is what we wanted to do was we wanted to ask what were the baseline capabilities of various cells in the body. So normally skin cells are instructed by their neighbors to have this sort of boring two-dimensional life as the outer surface of a tadpole, keeping out the pathogens, things like that. And so we are very interested in evolution and plasticity, and we wanted to know what would cells do in a completely different scenario. And so we simply took off a whole bunch of skin cells. So it's not just one cell, but it's a large number of cells that are already fated to be skin. We took them off of an early frog embryo, and we put them in a separate dish to give them a chance to kind of reboot their multicellularity and to see what would happen. Now, in principle, many things could have happened. They could have done absolutely nothing. They could have crawled off in opposite directions. They could have formed a flat monolayer, the way that cell culture often does. Instead, what they did was they overnight they basically assemble into a sphere, and one of the amazing things about them is that they have those skin cells have these little motile hairs called cilia. Then they usually are rotating these cilia around to keep a flow of water moving down the edge of the embryo to keep off the various pathogens and parasites and so on. And spread the mucus around. So these Xenobots, what they do is they use those cilia instead to move around. So they basically like little tiny oars. They sort of row against the water, and they do so in a coordinated fashion, and that enables the Xenobot to move around. Now, what we have demonstrated is a number of things. So these Xenobots They have self-directed motion, meaning that you don't need to pace them like other biobots. They move on their own. They have a variety of movement types. They can go in circles. They can go straight back and forth. They can navigate bends in mazes, without having to hit the opposite wall. They can regenerate. So if you take a pair of scissors and you cut them almost in half, they will come together and reseal back into their correct new Xenobot shape. And one of the most amazing things they can do is they do something called kinematic self-replication, which means that if you provide them with a bunch of loose skin cells in their environment, just sprinkle a bunch of loose embryonic skin cells in the dish, what the Xenobots will do is they'll run around in circles. By that motion, they collect they're sort of like little bulldozers. They collect these cells into little piles. And of course, we know what happens when you collect skin cells into little piles. They turn into Xenobots. So in effect, they're making the next generation of Xenobots. And when those Xenobots mature, guess what they do? They run around and collect new stem cells and make the next generation of bots. So it's basically kind of like von Neumann's dream, which is to have a machine that runs around, finds parts, and makes copies of itself out of parts that it makes elsewhere. Now, what's one thing that's super interesting about that is what we've done is we've keep in mind, we've not added anything to these cells. There's no genomic editing. There is no nanomaterials. We haven't really added anything. What we've done is something interesting and important. It's an important way of engineering. We've taken off some constraints. We've taken off the constraint of these other embryonic cells that normally keep these cells to a skin-like pattern. And we've also made it impossible for them to reproduce the way frogs normally reproduce. And what we see is that despite that, what they are able to do is find a completely new way of making copies of themselves that, as far as we know, doesn't exist anywhere in the animal kingdom. And so this is really important. And people often say, well, how come you didn't genetically engineer them? And sure, in the future, of course, we're going to put new synthetic biology circuits into them and so on. But the more important thing for now is to understand the native capability, plasticity, and ability to handle novelty of these cells in new environments without any changes. This is the same frog genome that is able to do all this stuff. And there's never been selection to be a good Xenobot, right? There's been selection to be a very successful frog, for sure, but there's never been a specific selection to be a good Xenobot. So that's very important. And then just the last thing, you said in your question, you asked about decision making. We can have a whole discussion about what it actually means to make decisions, because that's not at all obvious. But we've not yet made any claims about the cognitive abilities of these Xenobots, because that's very much under investigation still. So we are testing how, do they have memory preferences? What kinds of things can they learn? How rich is their behavioral repertoire? That all is still TBD. So we haven't made any claims about that, but we do know that they have a variety of novel self-directed behaviors, and we can talk about what it means to make decisions and so on.
Oh, that's amazing stuff. So do you have any theory as to how it can be that Xenobots are able to navigate their surroundings without having a nervous system?
Yeah, well, there's a whole field of research called basal cognition, which asks the question, what did life do before nervous systems came on the scene? And there are many different types of life forms that navigate their environment very successfully without a brain and without a nervous system. So there are, of course, single cell organisms like bacteria, paramecia, amoebas, and various other things. There are slime molds. We've done some experiments with slime molds where, for example, you can take a slime mold, put it in the middle of a dish, and some distance away, you put either one glass disc, and then you put one glass disc, and on the other side of it, you put 3 glass discs. And for whatever strange reason that we still don't understand why, the slime molds have a preference for heavier objects in their environment. And so what that slime mold will do, despite not having a brain or a nervous system, using a biomechanical sensor where it literally pulls on the substrate that it's sitting on and can feel the distribution of mass in its environment, it will preferentially go to the heavier mass. So it can sort of, at a distance, it could find the three discs rather than one. And these are just inert glass. There's no food. There's no chemical gradients there. So there are many examples, and maybe many people have wonderful examples. Of course, in plants, there's tons of examples in bacteria of creatures navigating their world. So biology has been solving problems in different spaces, including metabolic spaces, physiological spaces, transcriptional spaces, and anatomical morphospaces, meaning the distributed, all the space of possible anatomical configurations, long before it developed brains and muscles and started to use those same tricks to navigate three-dimensional space. But by then, a lot of this complexity was already in place.
Can we rule out the idea that consciousness is an emergent phenomena arising from the brain?
Well, look, I think it would take a really long time to get into that question properly, in the sense that we can't really say anything until we agree on a definition of what consciousness is, and it's unclear that we really have a good definition anyway. What I can say is that I don't typically say too much about consciousness per se, because it's extremely difficult to do experiments specifically on consciousness. What you can do experiments on very easily is cognition, behavior, and sentience, and those things, I think it's very clear at this point that those things do not require a brain. And the simplest way to the simplest way to think about that is, look, all of us made the transition from a single cell, right? So a collection of chemical reactions, basically, to a sentient being that's going to make claims about having consciousness and having beliefs and memories and so on. That transition was smooth and it was slow. There's no particular point in embryonic development where anybody can say, oh yes, there's the lightning flash that now turns this set of chemical signals into a creature that has actual sentience. Okay, there's no step like that. It's a very smooth process. So whatever it is, so basically the bottom line is, if we take developmental biology seriously, and if we take evolution seriously, whatever it is that you and I have, you can call it consciousness, you can call it whatever you like, whatever it is that you and I have, it developed very slowly and smoothly from a single cell state. So at a point where you certainly didn't have a brain, and in fact there's no time point at which you can say, bang, now you have a brain. Minutes ago you didn't, but now you do. So that just doesn't exist. The brain develops very very slowly. And if there is a theory of something very discrete that happens at a particular time point, I've never heard a convincing story about that. So I think all of these things are a continuum, and I think they, to some primitive degree, they exist all the way back. In fact, prior to single cells.
Yeah. Okay. What are some of the implications of your research, or should say not implications, applications of your research?
Oh well, there are many. So in our lab, we are trying to, for example, so any number of things. So first of all, just on the basic bioelectrics, the idea is that if you understand how it is that individual cells join into collectives that make decisions, so decisions about what shape they're going to build, right? The shape of the arm, the shape of the body, the shape of individual organs and tissues. The biggest thing holding back regenerative medicine is the lack of understanding of how cellular collectives make decisions. It's basically a problem in collective intelligence. And if we understood how that worked, we could solve pretty much all medical problems except for infectious disease. So birth defects, regeneration after injury, reprogramming cancer, degenerative disease, aging, all of those things would be solved if we understood how cellular collectives make decisions. And so one of the implications of our work is that if we understand how bioelectricity is used as a medium to implement the collective decision making of a group of cells, we could then come up with solutions to all of these problems I just mentioned. And we have, in my group, we've had proof of principle kinds of studies that show how you can fix birth defects, induce animals to regenerate their limbs, reprogram and prevent tumors, all those kinds of things. So basic regenerative medicine is, I think, one of the biggest sets of outcomes from all of this. Then, of course, there are things like, if we understood how biology solved problems and exhibited basal intelligence before brains came along, we could have architectures for machine learning that were not neuromorphic, that were not based on trying to copy the brain, which I think is very important. I think there's a whole set of pathways to general artificial intelligence if we could just step away from the architecture of the human brain. There are other applications, specifically of the xenobots. biobots in general are a great platform for understand for first of all for making useful synthetic living machines. So we're talking about applications in the environment, in the body, in exploration, in manufacturing, and so on. And then of course there's the whole idea of synthetic morphology, which is making novel constructs that never existed before, which teaches us a lot about plasticity. It teaches us about evolution and what it is that evolution actually learns during the course of a lineage, and how much plasticity and capacity cells have for novel types of configurations. And that sheds light not only on our origins in terms of our evolutionary origins, but more fundamentally what it means to be a sentient being that's made of parts, in particular cells.
Yeah, that's fascinating stuff. I'm curious, in the distant future, when you're able to, let's say there's a situation where you're able to sell morphology to human beings, and say there was a married couple who decided to change their morphology to become more muscular, would you expect their children to also grow up to be muscular as well without having the treatment?
Most things are not passed along through sexual reproduction, right? So
Yes.
with the exception of some really interesting work in transgenerational inheritance, and this is largely stress, as I understand it, it's largely about various types of stress that are passed on to your offspring. Most of the things you do to your body, including exercise and various modifications, are not going to be passed on. So I think if we were going to have offspring that were modified in some way, modifying your own body is not going to be the way to do it. You're going to have to give the embryonic process various signals to make things happen, right? And we know that works because right now many people are unfortunately exposed to signals that perturb embryogenesis in negative ways. So, smoking, alcohol, various drugs that are teratogens, stress, poor nutrition, all of those things impact the embryonic process negatively. There are an equal set of signals that I'm sure could be provided to impact it in positive ways. And at some point, I hope these things are within our control, so that we can all be healthy and enjoy more productive lives. But it's not going to be by exercising the parents. It's going to be by giving the embryonic process the information needed to make it as optimal as it can be.
Yep. Okay. What would be the best undergraduate course to enroll in for those listeners who'd like to contribute to research in your specific field within biology?
Well, this is actually a new field. It's a combination of disciplines that include biology, computer science, physics, engineering, and cognitive science. In fact, it even touches on things like philosophy of mind and so on. So I think the best thing to do to be in a good position to positively impact these kinds of research questions are to study broadly in all of those areas. So, for example, neuroscience, developmental biology, cell biology, physics, robotics, bioengineering, synthetic biology, good grounding in philosophy and mathematics. I mean, all of those things are helpful. I think this is a very interdisciplinary field. It is not going to be pushed forward by super specialists. There are of course very specific things you need to know, and specific in-depth classes you should take. But I encourage students to think very broadly, because increasingly the kinds of questions that could be answered within a single discipline are going to be used up, and we're all going to be facing some very broad questions that don't respect our sort of primitive divisions of the world into scientific disciplines, which we did that hundreds of years ago when we really didn't know much at all, and all of these distinctions are crumbling rapidly.
True. Yeah. If you could recommend one non-fiction science book and one book of any genre, what would be your recommendations?
Boy, that's really hard. There are so many good books. I suppose if I had to pick just one nonfiction book off the top of my head, I would probably choose the classic Gödel, Escher, Bach by Douglas Hofstadter. That's a classic. You can't go wrong with that one. So many things to think about there. And as if we're talking fiction, one of my favorite science fiction writers is Stanislav Lem, Lem. So he's written a bunch of things that I think are good kind of cognitive expanders to get you in the right frame of mind to think about what the space of possible beings and the space of possible minds looks like that we're all going to be living in some, in the not so distant future.
Well, when you say not so distant, like how far are you thinking?
Well, look, we already have. There are already humans that are part cyborgs, right? There are people who can drive wheelchairs with their minds and run prosthetic limbs and have brain-computer interfaces to various things. I think in our lifetime, in the next decade and 2 and 3, we're going to see a really radical expansion of what a standard human looks like because of these interfaces. So you could, you might have an interface to a camera that provides you with a human-like vision, but you might use that same port to instead get input that contains data from the stock market plus some NASA feed from Mars and maybe the output of some device in your laboratory that you're interested in, and you can get that into your cognitive apparatus the exact same way that you now process vision. So it could be directly plugged in as a form of the sensation. Same thing with actuation. People, when provided with prosthetics that don't mimic human. So for example, there was a nice paper where somebody had a prosthetic arm where the wrist was able to rotate all the way around, which normal human wrists don't do. No problem. They learn to use it very rapidly. And so again, you can imagine that, right? Part of the actuators of your body could be some sort of a wheeled contraption, extra pair of hands, and those hands might be on your body, or they might not be. They might be off in some factory somewhere. So that radical expansion of the typical sensory motor complement that humans have is already sort of being expanded right now, and in the future, all of the bioengineering technologies in terms of cyborgs and hybrids and various other ways to recombine evolved material, design material, and software. Every combination is a possible being, and we're going to see all of that stuff. It's going to make the sort of the opening scene of the original Star Wars movie, that iconic cantina scene. It's going to make that seem tame by comparison. That was a pretty good way to start thinking about this future.
Yeah, wow. We're in for a very interesting decade or two ahead.
Yeah.
Where can listeners best follow your work?
Couple places. So I have a website that's pretty up to date. It's drmichaellevin.org. And then I'm on Twitter with at Dr. Michael, at drmichaellevin.
Awesome. Michael Levin, thank you for coming on the podcast.
Thank you so much. Thank you for the conversation.